Hello and welcome to today's NPTEL lecture on Introduction to Mechanobiology. So, in the last class we have been started to discussing about cell migration. Okay. And the role of actin dynamics in driving cell migration. Okay. So, if I were to draw the individual steps for a cell to migrate. So, the type of migration that we are talking about is called adhesion dependent or the word mesenchymal is also used, mesenchymal migration. Okay. And cells like fibroblasts, epithelial cells, okay, all these types of cells which exhibit mesenchymal migration. So, what are the steps of cell migration? So, it always begins. So, if you have a cell resting on a substrate, so let us say these are my adhesions, it begins with, so let us say if I draw the position of the cell, it begins with protrusion, okay, protrusion of the leading edge. Okay. So, what you have a slight protrusion of the leading edge. Okay. So, the adhesion was here. Second, this protrusion is stabilized. So, stabilization of protrusion by formation of focal adhesions. Okay. So, you would have a new focal adhesion, okay, which I have drawn using red color. So, this is a new adhesion which stabilizes this adhesion. Okay. As a third step, you have translocation, where the cell is pulled forward, and the last step is retraction or de-adhesion of the trailing edge or the last portion of the cell. Okay. So, in this case, in this retraction, so rear adhesions are broken. Okay. So, what we were interested in was understanding how can we visualize protein dynamics during cell migration, okay, or specifically actin dynamics during cell migration. In this regard, I introduced this technique called fluorescence speckle microscopy. So, the idea is if you have a filament, okay, which is, okay, this is a filament where this blue color corresponds to fluorescent speckles. Okay. So, by tracking the position of these speckles and the intensity at these speckles, you can get information about flow and turnover. Okay. By turnover, I mean either polymerization or depolymerization. So, typically for fluorescence speckle microscopy, order 1 to 2% of monomers are fluorescently labeled. Okay. So, if you have a stress fiber, okay, if you have a stress fiber, where you are imaging using fluorescence speckle microscopy. So, instead of the intact fiber, you would see these dots which are kind of roughly aligned. So, you know that these are form parts of stress fibers. Similarly, if you have an adhesion, okay, you might have these dots which align at those adhesions. Okay, so, this is how you can track the dynamics of adhesions or actin at stress fibers using fluorescence speckle microscopy. So, using this technique, okay, so Claire Waterman Storer and Gordon's denuser they probed the role of actin dynamics in migrating cells. Okay. So, using FSM, the authors probed actin dynamics in migrating cells. Okay. So, what they found, so there were two things that I discussed in last class. If this is the leading edge, okay, so you would have two zones, okay, okay. So, what I have drawn are speckles, okay, the green dots, these are speckles. Okay, so, this is the direction of migration. So, what they found was exist two populations of speckles. Okay. All these speckles exhibit retrograde flow. Okay. Why retrograde flow? Because the speckle movement direction is opposite to the direction of motion or cell migration. But what you have are two zones. So, you can segregate this entire thing into two distinct regions of retrograde flow. Okay. So, you have one zone in which speckles exhibit fast retrograde flow, second one where move slowly. Okay. So, here retrograde flow is fast, here it is slow. Not only that, you also have, okay, so let us say depolymerization. Okay. So, the two zones, in the first zone where speckles move fast, you have two populations of speckles. The green one corresponds to speckles where polymerization is observed, okay, and the red ones signify depolymerization. Okay. So, what you have, so in essence the first zone, okay, first zone has high polymerization and high depolymerization, okay, plus fast retrograde flow. In the second ones, you have zone of slower flow and punctate turnover pattern. Okay. So, this zone, first zone is called the, so this zone is called the lamellipodia, and this zone is called the lamellum or lamella. Okay. So, what I also discussed was these two zones are molecularly distinct. So, in the, so let me call this big L and call this small l. Okay. So, in big L you have actin branching proteins like R23 and actin depolymerizing proteins like cofilin. Okay. In small l or the lamellum, okay, you have myosin 2 and tropomyosin. Okay. Myosin 2 and tropomyosin localize in this second zone. Okay. So, these zones are molecularly distinct and the retrograde flow as well as polymerization depolymerization dynamics is distinct. Okay. So, the question then arises that is it that these two zones are materially distinct or just there is a gradual difference in the behavior of the speckles. Okay. So, to answer this question, what the authors did was they took the edge of the cell, okay, and along at each point of the edge, let us take the strip A to B, okay, and along this tail you take normal to the edge, okay. So, what I have drawn are directions which are normal to the leading edge, okay, and we ask that how does polymerization and depolymerization vary along these lines. So, let us say this is line 1, so this can be line 2, so on and so forth. Okay. So, by tracking the intensity of speckles at various points along these lines, you can track what is the overall polymerization length, polymerization rate along that length. So, what the authors found is as follows. Okay. So, if I were to draw the polymerization rate, so what I find is, so this point corresponds to the leading edge. So, I am going from this point, I am going inwards, okay, or this point I am tracking inwards. So, what they found was the polymerization peak. So, let me draw with this. Okay. If this is the polymerization rate along this line. Similarly, so let me just point out this position of peak, okay, and this position. Okay. The depolymerization rate. Okay. So, because I am plotting the depolymerization rate, you will see that the depolymerization rate also, so I am plotting it in negative direction, exhibits a peak which is slightly off this peak, okay, then it drops and you have this line. Okay. So, this signifies the depolymerization. So, here the peak, okay, so this is the position of the peak, peak is right shifted. Okay. So, if I combine these two signals, okay, what I can get, I can plot the net assembly rate So, this is my net assembly rate. So, polymerization plus depolymerization, the sum of the two signals will give you the net assembly rate. So, what you find, okay, is a peak here. So, this positive peak, so let us say this is 0. So, positive peak significant of net polymerization, okay, followed by a negative peak which is basically peak of depolymerization and a minor peak here. Okay. So, what the authors realized was this zone, okay, and this entire zone. Okay. So, this entire zone where you have polymerization peak followed by the depolymerization rate is also the zone where the speckles are fast moving. Okay. So, this suggests that now, if you look at this curve, it looks like a sine curve, you have positive and you have almost symmetrically negative here. So, if you take the area of these two curves and sum them together, you will get 0, okay, suggesting that the material required for polymerization at the leading edge, okay, is provided by the depolymerization of filaments slightly backward. Okay. So, there is a providing of monomers, this depolymerization provides the monomers for the polymerization to occur. So, you have a polymerization event followed by a depolymerization event. Okay. So, suggesting that this raises the possibility that these two networks, okay. So, I have lamellipodia and lamella are materially distinct. Okay. So, which means, so when you say lamellipodia and lamella are materially distinct. So, if I draw this line here, let us say, let us assume this is the interface, this is the lamellipodia lamella interface. So, what this means, if these two networks are materially distinct, which means that within this first layer or the lamellipodia, the monomers required for polymerization are coming from the depolymerization of the filaments in the depolymerization zone within the lamellipodia. So, there are no monomer transport from the lamella to the lamellipodia. Okay. And so, this they also what they did to check whether this is true, they tracked the average lifetime of the speckles. And what they found was there are two classes of speckles, okay, fast moving and short living. So, these are the speckles which were present in the lamellipodia, and this makes sense because short living would mean that the turnover rate is high. So, the monomers get provided, they can get added at the leading edge leading to polymerization. And the second class was slow moving and long lasting. So, these were present in the lamella. So, the question that arises next is what is contributing, okay? What contributes to retrograde flow? Okay. So, you know previously that myosin 2, okay, this was enriched in the lamella, and what does myosin 2 do? It exerts, it pulls on stress fibers, on actin filaments. So, this generates contractile forces. If cell is anchored to the substrate. So, they did this experiment in which at the leading edge, okay, they treated cells with blebistatin. Okay. This is a drug which inhibits myosin 2 activity. Okay. So, it inhibits myosin 2 activity, and what they found was, so by doing perfusion experiments, what they did was, so let us say this is my leading edge, okay, and you have retrograde flow. So, you had fast retrograde flow here, followed by slow. This is fast, this is slow retrograde flow. Okay. So, this is without bleb. Okay. When you add bleb, these particles retain the retrograde flow, but internally all these particles retrograde flow is eliminated. Okay. Okay. So, you have retrograde flow nearly eliminated. Flow eliminated. So, this proves, so this point proves that when you have retrograde flow eliminated in the presence of blebistatin, which inhibits myosin 2 activity, it suggests that retrograde flow in the lamellar region is attributed to myosin 2 pulling along actin filaments. Okay. So, the filaments are pulled rearward, that contributes to the loss of retrograde flow when you add blebistatin. Okay. So, then what contributes to retrograde flow at the leading edge? Okay. So, to ask this question, so what contributes to retrograde flow at the leading edge? Okay. So, to answer this question, what the authors did was they treated cells with cytochalasin D. Okay. See, this prevents, this drug, it prevents actin polymerization. Okay. So, if this was my leading edge, let us say if this is my leading edge of the cell, what the authors did was they broke it down. Okay. So, they discretized the leading edge of the cell into small segments. Okay. So, each of these is a small segment. Okay. Let us say this is my segment 1 and this is my segment 40. Okay. And for each of these segments, so this is my time axis, okay, and along the edge, so this is position 1 and position 40. Okay. Along the edge, you can track the turnover, the edge displacement, okay. So, you have at each time, at each time step you plot these values and make a color map, you generate a color map. Okay. So, you plot turnover, you plot edge displacement and you plot network flow. Okay. So, let us look at how it looks under normal conditions. Okay. When a cell is migrating, so if you have a cell which is migrating in this direction, okay, so you will have three things, let us say. Okay. So, what I will do here, okay, in this graph, let us say this is turnover. So, before perfusion or without drug, okay, what you will see, you will have these, let us say, so as per our previous color schemes, green, okay, and these other zones, let us say, are red. Okay. So, this is different time snaps. Okay. So, this the way to interpret this is, at this point, let us say you start from some point 0, you call 0 point, okay. There is red is depolymerization. So, green is polymerization and red is depolymerization. Okay. So, the turnover says that along the edge there are some points where polymerization is happening, some points in the remaining points where depolymerization is happening. Okay. So, if I were to plot the edge displacement, let us say this is my edge displacement. Okay. At the position where I had polymerization previously, what I see, okay, and the rest points blue. Okay. Again I have red and so on and so forth. Okay. So, in this case, in this colored scheme, so blue is negative, red is positive and max. So, positive means, so the cell is migrating outward. Okay. So, where polymerization has happened, the edge has moved outward. Okay. That is my positive direction. Okay. Remaining points, so let us say where depolymerization has happened, the cell has locally the cell has moved backward. Okay. So, that is what you see. Okay. And for in terms of network flow, because it is retrograde flow, you will have blue throughout, but deep blue here and then lighter blue shades here. Okay. So, this is my retrograde flow, this is flow. Okay. So, a polymerization event is correlated with an edge displacement and correlated with retrograde flow. So, you have the following thing: polymerization causing edge displacement, and this causes retrograde flow. Okay. So, I stop here for today and we will continue from here in the next lecture. Thank you for your attention.